Welcome all. Very good to see all of you this afternoon. So we are indeed very fortunate to have Dr. Bill Bishai as our speaker today. Bill's career is truly studded with astounding achievements. Bill's research involves tuberculosis pathogenesis, novel drugs for TB, animal models for pulmonary infections and bacterial respiratory tract infections, and biomarkers to track disease. So a few words about his early career. He received his MD and Ph.D. degrees from Harvard Medical School. In 1989, his internship and residencies were done at Brigham and Women's Hospital at Boston, Massachusetts, and he received fellowship training in the Division of Infectious Diseases at Johns Hopkins School of Medicine. He did his Ph.D. with Dr. Murphy, who we have the great honor of having here with us. Indeed, thank you for coming. And then he was a Howard Hughes postdoctoral research fellow in the laboratory of the Nobel laureate Dr. Hamilton Smith. He joined Johns Hopkins faculty in 1994 and is currently professor of medicine in the Department of Medicine, Division of Infectious Diseases, and was the co-founder and continues to serve as the co-director for the JHU Center for Tuberculosis Research. One of his proud accomplishments has been to build an internationally recognized program of successful basic science in TB research at JHU with 6 faculty members, and the recruitment of Professor Jacques Grosse to the Johns Hopkins University in 2001, who truly made impactful contributions to TB drug development. So Bill's work has spanned many countries. From 2010 to 13, Bill served as the founding director of the Howard Hughes Medical Institute funded KwaZulu-Natal Research Institute for Tuberculosis and HIV in Durban, South Africa, where he supervised the construction of a $40 million research building and recruited 7 world-class scientists to its faculty. His work in South Africa led to new projects on the genomics of MXDR TB strains, small molecule biomarkers in human tissue, and T-cell immunology of TB in the peripheral blood and lung compartments of humans. He has authored over 280 papers in peer-reviewed journals and receives grant support from NIH, among others. He has trained a whole generation of TB researchers. Indeed, 4 of his trainees are now very successful faculty in his program. Needless to add, Bill has extensive service on international conference planning committees, study sections, editorial boards, review panels, etc., and 3 years on the WHO's Stop TB Partnership Coordinating Board. Please join me in welcoming Bill to give his talk.
Sarah, thank you for that very kind introduction. I want to start by telling you a story. An infectious disease doctor is walking down the beach on the Delaware shore, and he stumbles across a bottle, and he picks it up, and a genie comes out. And the genie says, "I'm here to grant you your one wish." And the infectious disease doctor says, "Wow, okay. Well, you know, in my Hopkins salary, I've never been able to take my kids to Europe. Can you build me a bridge across the Atlantic Ocean so I can show them the capitals of Europe?" And the genie goes, "The ocean is so wide and so deep. Give me a different wish." The infectious disease doctor is crestfallen and says, "Well, you know, in college I got a C- in history of medicine. Teach me the history of medicine." The genie says, "Back to that bridge. Do you want 2 lanes or 4? So now that I've sort of divulged my limitations in history of medicine, I have to say I was surprised when Sarah asked me to talk on the topic, but I'll do my best. I want to give you in the beginning a snapshot of what's happened over the last century and a half, 5 snapshots in time. And I'll take you back first to 1882. Prior to 1882, the etiology of TB was thought to be either genetics, bad living, the wrath of God, or some combination of the 3. But that all changed on March 24th, 1882, when Robert Koch, a country doctor, went before the renowned Physiologic Society of Berlin and displayed to them acid-fast bacilli in the tissues of humans and animals infected with tuberculosis. This was the beginning, the revelation that tuberculosis was an infectious disease. There was unbridled optimism. Koch became an international celebrity instantaneously. There was belief that now that we knew it was a microbe, a cure and a prevention would come along soon. And Koch went on to win the 1905 Nobel Prize. But for many years after that, not much happened. And it was difficult to cure and prevent tuberculosis. My second snapshot is 1902. Reality sets in. And I'd like to read some words from our own William Osler. Osler said, "Mr. Chairman and my long-suffering patient, inert fellow citizens, now what are we doing for the 10,000 consumptives who are living in our midst today? We are doing, Mr. Mayor and fellow citizens, not one solitary thing that a modern civilized community should do. And I tell you now, the TB condition in Baltimore is a disgrace to us, a city of 500,000 inhabitants. It's a story of desolation, want, and helplessness, and of hopeless imbecility in everything that should be our civic relation to the care of this disease." Osler was mad. And this was a reflection of the time. We knew it was a microbial pathogen, but there were no tools to intervene, no drugs, no preventions, and there was a feeling of helplessness. Fast forward to 1944. The man on the left is Professor Selman Waxman from Rutgers University, a soil microbiologist, and the man on the right is Albert Schatz, his Ph.D. student, who worked many months, 18-hour days, cross-streaking soil microbes from northern New Jersey against Mycobacterium tuberculosis, looking for zones of inhibition. And he finally found one, a streptomycin that produced the drug on the top right, streptomycin, one of the very first antibiotics, introduced in 1944. This was a time of great hubris. It was believed that this would eliminate tuberculosis. Waxman went on to win the 1952 Nobel Prize and wrote a book entitled The Conquest of Tuberculosis. Schatz, incidentally, was not recognized with a Nobel Prize. Even though Waxman believed in his book that tuberculosis would be eliminated, obviously, and the data I'll show you momentarily prove him wrong. But nevertheless, that concept remains in force today. Here, a magazine advertisement from Blue Cross Blue Shield saying, "One man defeated tuberculosis. Surely all of us working together can help keep healthcare affordable." My fourth snapshot in time is 1998. It was widely known then that the genome was about to be sequenced and released. There was optimism that the marriage of biotechnology and information technology would open the doors to our understanding of how Mycobacterium tuberculosis caused disease. But sadly, in the genome of 4.4 million base pairs, what we really found was lots of hypothetical genes and conserved hypothetical genes, and not a single obvious smoking gun. There were no toxin genes, no cytokine homologs, no homologs to known virulence genes from other pathogens. It was a time of appreciation of complexity. We began to realize this was a tough pathogen, is a tough pathogen, and we need to roll up our sleeves to figure out how it causes disease in humans. My fifth and last snapshot in time is 13 years ago with the recognition of extensively drug-resistant tuberculosis. These are photographs from northern KwaZulu-Natal, where I and some others in this room had the chance to work and study tuberculosis and HIV infection. And you see the crowded clinics and the open-air wards where, despite treatment, TB could be transmitted readily among these patients coming for help. And what was observed between 2005 and 2006, really to the very astute work of Anthony Moll, the second author on this Lancet paper, was that while Moll had been treating patients for decades in this small hospital in Tugello called the Church of Scotland Hospital, treating them successfully even when they presented with advanced tuberculosis and HIV by giving them the standard therapies for TB, something changed in 2005. 53 cases in that year came to him for help with HIV extensive TB co-infection. He put them on the standard antibiotics, and they failed to get better. Indeed, 52 out of the 53 succumbed to their infection within 3 weeks. Rapid mortality. When the strains from these 53 patients were sent to the microbiology lab in Durban, it was observed that they were resistant not just to 2 antibiotics, which is what we call MDR tuberculosis. Now it was 4 important antibiotic classes, so-called extensively drug-resistant tuberculosis. This sent alarm bells around the world. There was fear that extensively drug-resistant tuberculosis would expand and become a higher proportion of all TB cases. And many felt that we had actually undone the progress of the previous half century. Here's Mike Eisman, a famous TB clinician from Denver. In roughly 55 years, we have squandered our precious legacy of chemotherapy for these XDR cases. Darwin, though, would understand. So what will the next snapshot be? We're facing some monumental challenges. Now we have MDR and XDR. Are we going to go down as the generation who squandered the precious tools of the antibiotic era from the 40s, 50s, and 60s? So how do we get into this mess? As many of you know, TB is a bacterial airborne infection that's spread person to person. Annually, there are 8 to 10 million cases, new cases per year, and about 5% of those are multi-drug resistant tuberculosis, resistant to 2 important antibiotics. And that, you should know, takes 2 years to treat. And even so, here in the United States, it's difficult to treat with significant mortality rates and even higher mortality rates in resource-poor countries. About 0.5% of all cases are extensively drug-resistant, resistant to 4 antibiotic classes or more. That takes 2 to 4 years to treat, has significant mortality in the United States, and until very recently was essentially a death sentence if you happen to contract XDR TB in a resource-poor country. All of this happened under the umbrella of the WHO having declared tuberculosis to be a global health emergency way back in 1993. Well, what's going on? TB rates have been rising steadily through the recorded epidemiologic history. We're setting record highs worldwide. Some would say we're plateauing. But in contrast, here in the West, we're setting all-time record lows. And look, beginning in the recorded epidemiology from England and Wales, rates were coming down beginning in 1838, well before Koch's discovery of the tubercle bacillus and well before the chemotherapy era in the 1940s. So it makes the point that tuberculosis is a disease of poverty. It was controlled really due to the advances of the Industrial Revolution in Western societies. And it makes the point that we desperately have dropped the ball with new tools. If we're going to succeed in these resource-poor countries, we need better diagnostics than the acid-fast smear that Robert Koch introduced in 1882. We need better vaccines than the BCG vaccine introduced by Calmette and Garan in 1921, and we need better drugs. I firmly believe that science holds the key. We can develop those tools with a better understanding of the pathogen. But let me give you a real-life example of why the tools that are working here in developed societies fail in resource-poor settings. Meet Lindo, a widowed mother from rural KwaZulu-Natal. She's been coughing for 2 months and finally decides to walk 3 hours to get to the clinic. She has a low-grade fever. She's had some weight loss and weighs 150 pounds now, but doesn't know anybody who has tuberculosis. She waits in the waiting room for 2 hours, and then a clinic worker gives her a sputum cup and asks for the sample and says, "Come back in 1 to 3 days after we do a smear on your sputum." Well, Linda says to herself, I don't think I can come back to the clinic. I have to tend to my children, and it's a 3-hour walk. The doctor's saying to himself, our lab is overworked, it's hard to get good smear results, and the nearest x-ray is 200 kilometers away. So this is the dynamic that plays out day in and day out. A person in a resource-poor country starts to cough. She sees a traditional healer. She's given an herbal remedy. She finally sees a doctor. She takes a cough suppressant. She then sees a religious leader, performs a ritual cleansing, and finally comes to a clinic where the diagnosis of tuberculosis is possible. Will it happen on the day that that clinic visit occurs? All too often not, and the disease goes undiagnosed with almost certain spread to the children and expansion of the disease. We need to be better at having rapid diagnostics and blister pack therapeutics that work rapidly. It's imperative that we diagnose TB in the clinic at the first visit at the point of care, not after the cat is out of the bag in hospital settings where the patients have advanced disease. But there's a second reason we fail to control TB. There's a mismatch between the disease epidemiology and the resources mobilized. TB is now the #1 single infectious killer on the globe with 1.3 million deaths per year, and it makes the top 10 of all causes of death. Number 2, as a single infectious disease killer, is HIV/AIDS with 0.9 million deaths per year, and number 3 is malaria. But if you look at the proportions that are spent by funding agencies, in this example, the Global Fund for AIDS, Tuberculosis, and Malaria, there's not congruency. The number 1 expenditures are on HIV/AIDS, number 2 is malaria, and number 3 is tuberculosis. We need to mobilize resources better, and a major problem, and a reason that it's been difficult to do that, is that the epidemic is invisible in the West, yet a global health emergency in the rest of the world. So how have we failed to get into this mess? Failures to diagnose and treat. The diagnostics until recently have required 4 to 8 weeks for culture. Mycobacterium tuberculosis is a slow-growing pathogen. And so to get the definitive diagnosis is time-consuming. Short course chemotherapy is 6 months. It's kind of a joke, isn't it? And prevention with the TB vaccine way back from 98 years ago has limited efficacy. And I would also posit that we've failed as scientists to appreciate the complexity to yield more modernized tools for diagnosis, treatment, and prevention. So what I'd like to do in the rest of my time is to give you a brief overview of the disease and then try to convince you that we are in fact working our way out of this mess and we're not guilty of continued hopeless imbecility. And I'll give you 3 eras of progress: the early advances here at Johns Hopkins, the antibiotic revolution with sanatoria converting over to short course chemotherapy, and then some very recent modern advances. So tuberculosis is a multi-stage human disease. It starts with exposure, close contact, usually defined as being in a closed airspace with limited circulation for at least 4 hours. And if you're in such a situation, like perhaps on an airplane next to someone coughing, you have a 30% chance of getting infected with Mycobacterium tuberculosis. The majority of the time after infection, the host immune responses contain the infection but don't eliminate it. That's so-called latent TB infection, diagnosed by the tuberculin skin test or modern blood test called interferon gamma release assays. Individuals who have latent TB, I happen to be one of them. To my surprise, after I finished medical school, I had a positive TB skin test, carry approximately a 5% lifetime risk of reactivation unless there's an insult to the immune system, chemotherapy, HIV, chronic steroids. Those increase the rates of progression to active disease. It's possible to be infected and then get primary TB. That happens 10 to 40% of the time. And then with active disease, an understudied part of the disease cycle is the physiological changes that lead to caseous granuloma formation in the lungs and tissue, and then the cavitation of those lesions, cavities being a very efficient way of transmitting viable organisms into the airways to lead to further exposure. If you look at the global epidemiology, approximately 1 person in 1,000 has active tuberculosis, with approximately 8 million people in an 7.5 billion planet population. And those individuals are capable of transmitting to the approximately 2/3 of the population who are naive. It's estimated that about 1/3 of the population has latent tuberculosis, and that's an important reservoir for future reactivation. So if we plan to eliminate tuberculosis, not only do we have to take care of these active disease patients in blue, there will be a long-term effect of these billions of individuals who have latent TB. We diagnose latent TB by the tuberculin skin test or these blood test interferon gamma release assays such as T-spot TB or quantiferon, and we diagnose active TB, at least until recently, by the smear, the microscope test that Robert Koch introduced, X-ray, solid culture, and liquid culture, but I'll mention some innovations in a moment. So, much has been written about the pathogenesis of tuberculosis. Two of my favorite authors are Arnold Rich, a Hopkins man, the chairman of the Department of Pathology for many years, who wrote a very important text called The Pathogenesis of Tuberculosis in 1994. And then my friend and colleague, the late Arthur Dannenberg, who was here for 50 years, wrote another important monograph, The Pathogenesis of Human Pulmonary Tuberculosis. But I found over the years that a better way to teach students about the pathogenesis of tuberculosis comes from this cartoon series called Huber the Tuber by Harry Wilmer, who issued this book in the mid-1940s, sponsored by the National Tuberculosis Association, which is now the American Lung Association. So let me show you a little bit about what Wilmer had to say about the pathogenesis of tuberculosis. Here's Huber the Tuber, the villain. Clearly some post-World War II imagery going on here. And Huber gets to enter the lungs through the trachea into the promised Lungaland, where there's a great battlefield, and there's the final celebration, and there's an entrance into the pleural space and lots of exciting things. In fact, up here, I believe, he meets his girlfriend Bovie, an allusion to Mycobacterium bovis, cattle tuberculosis. Well, the first thing that happens is the initial deposition from a droplet that has Mycobacterium tuberculosis in it. Here you see Huber and his buddies beginning to proliferate rapidly in the alveolar space after the initial infection. The next thing that happens is that they enter the lymphatics and disseminate using the lower lobe L, lymphatic rapid transit system. And then they start building those little granulomas that I mentioned. And here's Huber using blood calcium, mold, tissue juice, and plaster to make these calcified granulomas. And then this host is containing the infection, getting latent TB. So here's Huber going latent, falling asleep to the reading of the Magic Mountain by TM. And it's a Gone complex with the sign Chief Gone. That's a radiologic hallmark of latent tuberculosis, and nerve pull for emergency exit. Well, at the end, where there's reactivation tuberculosis with granuloma breakdown and cavitation, here's Huber and his girlfriend Bovie breaking down the extracellular matrix, developing a cavity, and going on to lung destruction. If you ever want to look at this, I have the whole book in my office. It's a good thought, you know? They're never too young for this stuff. So let me tell you about some of the things that Johns Hopkins has to be proud about with respect to undoing the hopeless imbecility of tuberculosis control. I mentioned Osler, who actually trained under the great German pathologist Rudolf Virchow. He was our first physician-in-chief and called TB control hopeless imbecility. He and Welch had a number of epidemiologic and TB control research projects ongoing here in Baltimore, Welch being the founder of our own School of Public Health. Also, many of you will be aware that the great epidemiologist Wade Hampton Frost, who was the chairman of the School of Public Health's Epi Department from 1919 to 1938, and then the dean from 31 to 34, made great advances in epidemiologic planning and trial design, and is credited with having developed important epidemiologic methods for cohort analysis using a TB cohort here in Baltimore. Richard Riley, who is chairman of the School of Public Health's Environmental Health Sciences Department, is credited with having demonstrated in an elegant experiment the nosocomial transmission of tuberculosis. He did that in the Baltimore VA hospital at a time when it was at the intersection of the Loch Raven and the Alameda, a couple miles north of here. And cleverly and elegantly and beautifully, he simply put guinea pigs, which are highly susceptible to MTB, in the ventilatory system of the hospital. A set of guinea pigs over the tuberculosis ward in that ventilatory system, a set of guinea pigs over the general medicine ward. And lo and behold, only the ventilatory system of the TB ward was able to infect the guinea pigs. And this ushered in important advances in prevention of nosocomial infection, and is the reason we have to wear those white spacesuits when we go see a TB suspect in the hospital. I mentioned Arnold Rich. He was the chairman of pathology from 44 to 58, wrote that monograph that I mentioned, 1,028 pages. And he's also credited for identifying what's well known in pathology as the Rich focus, a granuloma that straddles the meninges and upon rupture leads to TB meningitis. John Hanks, again from the School of Public Health, who was the director of the Leonard Wood Memorial Research Labs in the pathobiology department over in the School of Public Health, was a leprologist who spent his life trying to cultivate Mycobacterium leprae, the causative agent of leprosy, a close relative of Mycobacterium tuberculosis. His and the efforts of others have remained unsuccessful. M. leprae is one of the few uncultivable human pathogens. But in the process of trying to grow it, he developed the famous Hanks balanced salt solution, which I'm sure most of you in this room have used. George Comstock was a professor of epidemiology for 51 years here at Johns Hopkins, known to some of us in the TB Center and one of the members of the TB Center until his death in 2007. He was a giant in TB control in the United States. He was the first to use BCG in the United States in the South from 47 to 51, and was critical in the decision not to use BCG in the United States, but rather to rely upon tuberculin skin testing as the main tuberculosis control tool. And later on, he spearheaded community-wide preventive therapy in Alaska among the Alaskan Inuits, who were afflicted with heavy rates of tuberculosis due to the gold rush to Alaska in the 1950s. White people coming up from the West Coast were infecting the Inuits, who were highly susceptible. And Comstock said, let's give an entire village isoniazide, no advanced testing, and let's not do it in another village. And he showed very clearly isoniazide had benefits in preventing the emergence of active tuberculosis, which is now why we treat individuals with latent TB with isoniazide. And then finally, my friend and colleague Arthur Dannenberg, who died last year, professor in the EHS and a teacher, taught many of us personally and developed the rabbit model of cavitary tuberculosis, which is ongoing today. Well, let me tell you a little bit about some of the success stories from the middle part of the last century. And I'd like to do that by telling you a real case. This is the case of Dr. J.G. And he, at the time, was a 23-year-old medical student in France. 2 months prior to being posted to the pediatric TB service, he had a normal chest x-ray. But then on Christmas Day in 1952, he noted fatigue, anorexia, and weight loss. And then New Year's Day, shortly thereafter, had hemoptysis with bright red blood, which clinicians recognize as a sign of tuberculosis. He had a repeat chest x-ray and now had a 2-centimeter right upper lobe nodule. Even though his sputum was smear culture and culture negative, they knew it was tuberculosis. He was hospitalized and took the 3 leading antibiotics at the time: isoniazid, streptomycin, which, as some of you, as one should appreciate, is an injection in the gluteus maximus daily, not much fun, and para-aminosalicylic acid, which has a lot of GI side effects. He took that diligently, but had to stop medical school and had home rest therapy. But his lung pathology worsened, and now he had a cavity. So his family sent him to the sanatorium in the Alps, the Saint-Hilaire-du-Touvet in Isère, France, where he lived in conditions like this, having to be at bed rest for 23 hours a day. Your meals were brought to you. You were rolled out onto the veranda on your bed to take the fresh air and the sunshine. And it was believed that fresh air and sunshine would bring about the cure of tuberculosis. Well, despite being at the sanatorium, his diagnosis now evolved to tuberculosis unresponsive to therapy, and he had persistently positive cultures and had to have this total bed rest that I mentioned. Finally, after failure to improvement, the doctors told him, your best years are behind you, but we're going to try one last thing. We're going to try to surgery to remove the worst part of the infection, the right upper lobe, and then another 3 months of bed rest. That did the trick. He improved, he went off antibiotics and was discharged now 2 years and 9 months later, having to be out of medical school for that entire time. His dream was to become a surgeon. Now that was no longer feasible. But interestingly, a co-patient of his was the famous Georges Kennedy, who was a professor at the Pasteur Institute, and they played bridge together at the sanatorium. And Kennedy, as he watched Grosse's dreams of becoming a surgeon fade, said, come with me, come to Paris, I'll teach you microbiology and animal models of tuberculosis. And so Jacques Grosse, JG, shown on the right, went to Pasteur and later on became an independent scientist at the Pasteur and conducted decades of seminal research on better antimicrobial regimens for tuberculosis. And then, as Sarah mentioned, to our delight, at the age of 70, Jacques said, let's write a grant together. And when we got it, he said, I'm coming to Baltimore, and he did. And even though I thought he was going to check into the Marriott and eat some nice dinners and go back after a week, he said, Bill, where can I get an apartment and where can I buy a car? And he stayed until 1987. He stayed for 15 years, was appointed professor of medicine, and continued his very illustrious career with a second career here in Baltimore. And in the process developed short course treatment for Buruli ulcer and showed that the antibiotic clofazimine may be a very important agent for the treatment of TB. So the sanatoria are now a thing of the past. Thanks to these 10 or so antibiotic classes that came in the 1940s through the 60s. And now we have short course chemotherapy for TB with 4 drugs for 2 months and 2 drugs for 4 months. The drugs abbreviated H for isoniazid, R for rifampin, Z for pyrazinamide, E for ethambutol. And we've even honed it down to 14 daily doses and 48 twice weekly doses for a total of 62 doses. And these are to be given by directly observed therapy. The cost of the pills is several $100s. The cost of a healthcare provider going to the patient to give the directly observed therapy is at least $15,000. Nevertheless, rich countries like ours can do the trick. But look at how much a patient has to take. Nearly 1/4 kilogram of antibiotics just for simple drug-susceptible tuberculosis with short-course chemotherapy. The dream of many of us is to come up with something way shorter, like we treat other bacterial pneumonias, where you could hand the patient a blister pack, maybe a month's worth, maybe 2 months, to shorten the course of chemotherapy. Think about that patient from KwaZulu-Natal. If had been given just a blister pack and told, just take these, you'll be cured, things would be dramatically different. The last thing I want to tell you about are some recent modern advances. And as I said before, we need to work on diagnostics, treatments, and preventions. And here are some things that have really gone well in the last 20 years. In 2010, the company Cepheid on the West Coast introduced Gene Expert for tuberculosis. It's a device about the size of about the size of a microwave. This was the one that can take 4. You put the sputum into this cartridge, and through the wonders of microfluidics and PCR, it tells in 90 minutes to 2 hours whether yes or no there's Mycobacterium tuberculosis there, and yes or no is there rifampin resistance there, rifampin being a key antibiotic. And its sensitivity is almost as good as culture. So Gene Expert, which cartridges cost around $20, has been launched in many parts of the world. We even use it here. It's valuable in TB management here in this country. But nevertheless, it has not fully achieved point of care. It's usually used in overseas settings in reference centers. So Lindo, the widow from rural KwaZulu-Natal, would still have to have her sputum sent to a reference center. The test would be done quickly, but the data would still come back after her clinic visit. So we haven't quite achieved point of care, even though Gene Expert is a very significant advance. And there are 2 new antibiotics. In 2012, Janssen Pharmaceuticals developed bedaquiline or Serturo, and 2 years later, Otsuka received EU and approval in other countries for its drug delaminid, a nitroimidazole drug. So one would think that with 2 new drugs, multi-drug resistant tuberculosis, tuberculosis resistant to 2 antibiotics, would be eliminated. But sadly, that's not the case. These drugs were fast-tracked approved for extensively drug-resistant tuberculosis. They were studied in patients with XDR TB, and the endpoint was culture conversion or clinical improvement, but not cure. So they were launched only for XDR TB, and we still don't have full clarity on how to use them for the vast majority of TB patients, MDR TB patients and XDR TB patients. Lots of clinical trials are in the works, and I think we'll have it straightened out thanks to the work of many here and elsewhere, but it didn't come overnight, and we may have had to wait a decade to get clarity on how to deploy these 2 new drugs. And what about a vaccine? Well, there have been some very exciting developments. Here was a trial published last year looking at individuals with latent TB being vaccinated with an experimental vaccine, a fusion protein of M. tuberculosis plus an adjuvant, with the goal of preventing progression. So these patients are already infected, they have latent TB. Did this agent work to prevent progression? And indeed, it was 54% effective, only 10 cases in the vaccinee group and 22 in the placebo group. So for the first time, we have a hint that it's possible to give a vaccine to patients with latent TB that will block progression. Similarly, one would also want to prevent infection. In that first part of the circle I showed you, can we block initial infection with a vaccine? And again, there have been some promising findings. Here was a study looking at another MTB subunit vaccine, a recombinant protein with an adjuvant. And these were adolescents from Africa who had already been vaccinated as infants, who were given either BCG or this recombinant vaccine to look what would happen in revaccination. Interestingly, the recombinant vaccine didn't protect, but for the first time we appreciated that BCG revaccination actually was valuable. It showed statistically significant 45% protection. Many say BCG is now making a comeback. Maybe we need to give it every 10 years. Maybe that will be the trick. And here's some very interesting data just published in Nature Medicine by Frank Verrick and his colleagues in Holland. Verrick was studying the use of BCG intrabronchially or intradermally, as it's usually given to humans. And so he BCG vaccinated monkeys either intrabronchially or intradermally, waited a number of weeks, and then in a very interesting model, infected them weekly 8 times, each time with 1 CFU of Mycobacterium tuberculosis. So it's ultra low dose challenge, but in the intrabronchially treated monkeys, 3 out of the 8 monkeys got no infection, even with 8 repeated exposures to 1 CFU. So again, an indication that maybe with further research we will, and further modes of vaccination, will be able to prevent TB infection. So there has been progress. Diagnose and treat. We have a new diagnostic, a 90-minute one called Gene Expert. We have 2 new drugs from 2012 and 2014. For prevention, we have agents that show promise. They don't go all the way for both TB progression and also for TB infection. So the outlook isn't so grim. I don't think we're going to go down as the generation that squandered the precious legacy, or the generation of hopeless imbecility. I think the future is bright, but much more has to be done. Before I close, I want to thank the TB Center. It's been a privilege for these 20 years to work with talented people like you. These are just some of the faculty and staff, but there are hundreds of others, and they are really my truest mentors. Thank you very much.
You know, in a kind of a crazy kind of way, cancer mimics this. In other words, you get cancer, right? And you're a woman, and you get resected, you get chemotherapy, and then 18 years later, you get a lesion in your lung, okay? So this idea of hiding out, so the concept of even lung gavage, I mean, if they're hiding out in the lung, why can't you do some kind of, essentially topical therapy to them, as opposed to having to even do systemic therapy? Because isn't what you're telling us, let me back up. They can go to other places besides the lung. I can remember a famous patient here at Hopkins who apparently had tuberculosis of the prostate, was told he had metastatic prostate cancer, and was given androgen ablation and was cured.
It was good news that he had tuberculosis,
but
right?
They never really told him he had tuberculosis. That's the other. So the idea is, besides the lung, where does it go?
Yeah, so one thing Art Dannenberg taught me, which is in the old literature, is that on that initial exposure, there's proliferation for 3 to 4 weeks before acquired immunity kicks in and starts the containment process. And during that 3 to 4 weeks, there's a road trip from the original lesion through the lymphatics, thoracic duct, right side of the heart, rest of the lungs get seeded, then the systemic circulation, rest of the body gets seeded, all before the acquired immune response wakes up. So that's how patients get extrapulmonary TB. It's in that first month. Even if they're going to contain it, there are thought to be little rests of M. tuberculosis elsewhere besides that Ghosn complex that you might see on the X-ray. So it's probably not as simple as there's just a couple of bacilli in that calcified granuloma. There's been some exciting research that maybe it's in hematopoietic stem cells, that stem cells are long-lived phagocytic cells. Maybe that's where it's hiding out. Maybe it's in the bone marrow. And there's been some evidence in that direction. But we don't really know where latent TB is, and there's no such thing as a test tube of latent TB. You can get it to go quiescent, but there's no evidence that a quiescent culture, a starved culture, if you will, is the same as the physiology of the bug in those latent lesions.
You speak about development of biomarkers. I belief that that would be for early diagnosis or for progression. So would you just touch on that for us?
Yeah, we compare ourselves to HIV all the time. We're the third bunny over the fence with respect to funding. If you get HIV or if you have a new HIV drug, you give it to patients alongside a control regimen, and you follow the CD4 count and the viral load. With TB, patients go from sputum culture positive to culture negative after about 8 weeks, but they have to be treated for 6 months. After they go culture negative, we're in invisible space. There's no biomarker there for whether the patient is going to be cured or not cured. 95% of drug-susceptible TB is curable. There's 5% who are not. And then to find out if your drug regimen succeeded or failed, you have to follow those patients for another 2 years, looking crudely for the reemergence of tuberculosis. So we have this blind space where we don't know, we don't have very good, we have zero in the way of microbial biomarkers. There are some people who believe that transcriptomic signatures may be valuable. There's a type 1 interferon response which seems to precede the onset of active tuberculosis. Whether or not you can rely on host biomarkers when many patients have HIV-TB co-infection is yet another question. So and I think, the pharmaceutical companies all say, well, it's $100 million to do these phase 3 trials because you have to follow patients for 3 years. Give us a biomarker, the cost will go down. So I do think that's a high priority.
Why is the rabbit the choice model?
Yeah, so mice, we use them all the time, but when you infect a mouse, the counts go up by about 4 logs, and then there's a plateau. Some people who I don't agree with call that plateau containment. Ultimately, the mouse will die. You infect a guinea pig, same thing happens, the guinea pig will die. You infect a rabbit, there'll be an expansion of the population of bacteria and then a decline, and rabbits will go latent. So it's the smallest animal that can get latent TB. Sadly for us, we have a great rabbit model. The Gates Foundation wants non-human primates. And the 3 or 4 labs in the country that can do TB with monkeys are getting handsomely funded. Rabbits also get beautiful cavities, which mice and guinea pigs don't. So there's a problem with the pathology of our rodent models. I guess guinea pigs aren't truly rodents, but so I do think the rabbit model should be kept alive because the pathology is much closer to human.
This is the history situation. For a lot of us in this audience, they won't remember, but Baltimore was like an epicenter. In other words, we had a lot of sanitariums. And why, in retrospect, we've got this great school of hygiene and public health with the cohorts, is it just that it was a dirty city? What's the significance of Baltimore being such a high level of TB?
Yeah, it was thought to be due to the port, being in its heyday in the middle part of the last century. And that said, I don't know all the demographic reasons why we had so much, but we were near the top. But Schaefer, when he was mayor, decided that it was an embarrassment. And he hired a Vietnam vet pulmonologist, whose name is escaping me, to be the head of TB control right here on Caroline Street, where the Eastern Chest Clinic is. And that individual said, we're going with directly observed therapy. So we instituted clinic-based DOT, and then 2 years later, a community-wide. Nurses actually go to the patients. And then we became the poster child for TB control. We had rates that came down way faster than Atlanta, the home of the CDC. Denver was close second. So it really goes back to Schaefer saying, we need to get on top of this. But John, I don't know the ins and outs of why we had so much in the first place. Nate?
Do you still have no idea what the toxin is or the pathogenic receptors for binding?
So there, TB is an intracellular pathogen, and if you look at our colleague intracellular pathogens like Salmonella, Listeria, Chlamydia, they secrete proteins into the cytosol which take over the signal transduction within the cell, and they basically remodel the macrophage into a comfortable environment. there's evidence now of maybe half a dozen such secreted molecules of M. tuberculosis. With Salmonella, their list is about 400. So we're behind. We blame it on the slow-growing nature, but I'm not sure that's a full excuse.
So if there are no more questions, I beg your indulgence for a few more minutes, in that you might be all wondering how these wonderful series come about, and as you just enjoyed Dr. Bishai's talk, and we have a series of seminars that are organized by our cancer center. So all this doesn't happen just like that. I come and speak to you and I present the speakers, etc. But the less appreciated people are those who organize these and do all the legwork for this. You have often seen Sam Lee outside herding you through, taking your slips, etc. And today I'd like to honor him by asking him to come and present the plaque to Dr. Bishai. Would you please? And do you have, yes.
Sam asked me a month ago what kind of sandwich I wanted.
See?
He's very organized.
That's right.
You gotta ask the important questions. Absolutely.
This way, we need a picture. Please come upwards.
Thank you very much.